Okay, then let's see what we can get through here. What I'm wanting to do today is to finish up this part on eukaryotic gene regulation, and that's what we're doing with chapter 16. We've already gone through a portion of that, getting up to this idea that while we have the alternative splicing, so you're able to actually, with cells, produce different proteins from the same gene. And this was the example I had with that, with this tropomyosin protein that shows up in two different kinds of muscle. And it's slightly different versions of that protein, depending on the type of muscle. So we had all that. And then there's other aspects of gene regulation to look at. Then we'll spend most of the rest of the time looking at bacteria and viruses. So that definitely gets interesting from a disease standpoint. Obviously, bacteria and viruses kill a lot of us, and we'll see some of how it is that they do that. So we'll spend some time with those, and then we still have a group discussion to do. That'll be the last one of those we're doing this term. I wanted us originally to do 9. Instead, we're ending up with 6. Because of the various snow days and interruptions, I had to cut some of that out. Well, we've already looked at transfer RNAs, ribosomal RNAs, messenger RNAs, all these little prefixes that go in front of the letters RNA. Here's another one, microRNAs. These are used to regulate the messenger RNAs. So what you'll have is perhaps a long messenger RNA with 1,000 or so nucleotides in it, and that is a single strand. But then the cell can make a microRNA which base pairs with just a portion of the mRNA. So if the mRNA is like this, the microRNA might be this long and attach part of the messenger RNA. And what it can do is regulate the reading and the lifespan of the messenger RNA. So the cell can have messenger RNAs floating in the cytoplasm and, for instance, temporarily deactivate them if it wants to have the messenger RNAs available for making a protein. So it's already done some of the steps that it needs to do to make a protein, but it doesn't want to make the protein yet. Then it can produce a microRNA that will bind to that messenger RNA and just put it on hold for a while, deactivate it until it's ready to use the messenger RNA. So if you had, say, a sequence of, AAAA on the messenger RNA, the microRNA could read U and pair up with that. It would be more sophisticated than that, but that's the idea. So we end up making thousands of different varieties of these microRNAs in addition to the different messenger RNAs that we make. This can block translation as long as the microRNA is there, preventing the ribosome from being able to bind correctly to the messenger RNA and start moving through it to make protein. Also, some of those can actually trigger digestion breakdown of the messenger RNAs. So these microRNAs can be used to signal that the messenger RNA needs to be broken down and gotten rid of. So different functions here. If you already have a particular messenger RNA in the cell is able to make a microRNA, binds to a portion of that, and targets this messenger RNA, possibly to be broken down, or else to be inhibited to prevent translation from occurring for a while until the cell is ready for the translation to occur. So this is part of the sophisticated control of gene expression. Remember, gene expression is taking us all the way from having the gene in the DNA to having a functioning protein. There are many different points of various processes in between that can be sped up or slowed down. Here's an example where we slow down a process. And then, even after that messenger RNA is active in the cytoplasm, well, as I said, you can have the messenger RNA broken down. It doesn't last in the cytoplasm forever. Also, controls of when the ribosomes are actually beginning to work to make the polypeptide, which is part of what we were just seeing. but also the proteins themselves often need some additional work done on them before they're fully operational. Just making the polypeptide does not automatically mean you have the protein, and I'll show you an example of that. And then the proteins themselves don't have to last forever in the cytoplasm. Sometimes they're broken down after a time. So there are lots of things that basically can dial up and dial down the amount of gene expression that is going on. Here's one. If you are starting with, say, the pre-mRNA, say you just copied a gene from end to end. So now this is messenger RNA coming straight off the DNA molecule, introns and exons, everything included. It gets processed. You end up removing the introns, of course. And one thing that gets added onto it is what's called a poly-A tail. That is a long sequence of A bases, hundreds of them, but it's variable. And this poly-A tail has a lot to do with how long the messenger RNA remains viable in the cytoplasm. The messenger RNAs can eventually be broken down by enzymes, and the poly-A tail has a protective aspect to prevent that. Generally, the bigger the tail, the longer the messenger RNA lasts. So the cell has the option of putting on poly-A tails that are longer or shorter, and basically giving the mRNA either a longer or a shorter lifespan before it's digested. So that is something else. The poly-A tail can vary in a grid between about 50 and 250 bases long. It's just a long series of A bases or nucleotides. So the messenger RNAs in various ways can have different lifespans, and that relates to how many copies of a protein you're going to make. Then the protein itself. This is how we get insulin initially. It's a single polypeptide, about 80 or so nucleotides long. It curves back on itself. It forms 3 of these covalent bonds between the cysteine bases, where you have the sulfurs lining up with each other, and they form covalent bonds. So you end up getting 3 of these connections in there, but with a single polypeptide. But this is pre-insulin. Imagine what your pancreas is having to do. It is working moment by moment to try to stabilize blood sugar. Now imagine you went and just ate a big piece of cake, and now there is a ton of sugar going into your bloodstream. Your pancreas wants to immediately release insulin to counteract that. Now, if it has to go through all the steps of transcription and translation, it's going to take a while before it can release insulin. Meanwhile, blood sugar goes too high and it has toxic effects that build up over time, like with diabetes. So what the cells in your pancreas are doing is they're going all the way through transcription and translation. This is past where the ribosomes have made the polypeptide. We're past that stage. This pre-insulin is stored inside of the pancreatic cells, and then when the pancreas detects blood sugar going up, it cuts away this portion of the polypeptide, producing the actual insulin, which then is released. So the pancreas very quickly gets on the job of releasing insulin. Otherwise, it would take longer to go all the way from the gene through transcription and translation to get to this stage. It's already done 95% of the work of making insulin at this point, and so instead of taking an hour to make more insulin, it just takes a few minutes. So protein activation, that's what's going on here. And it means that the cell has had to go and do a lot of work up front, but it's going to use the insulin eventually, and this is something where it makes the most sense to have it almost ready to go, virtually in the finished state, and then it's out in the bloodstream and it's doing its thing in almost no time. So one textbook, not yours, has this diagram where it's looking at all the different stages that are involved in going from gene to protein, and considering all these different stages that we've been talking about being like a series of pipes with valves that can open or close. So if you think about water trying to get from a reservoir eventually to coming out a faucet in your sink, it has to go through a number of points of machinery along the way. And valves that could be opened up, allowing more flow, or closed, restricting the flow. We have all these different things here going back to the structure of the chromosome, and then how you end up getting the transcription, the editing of the messenger RNA, removal of introns, splicing of the exons, the tail, the size of the poly A tail affecting what happens, export of the messenger RNA eventually being broken down, the protein being activated at selected times, protein maybe eventually being broken back down to amino acids. These are all control points in gene expression. And so it's a very sophisticated process and normally very well regulated. Obviously, there are things where it goes wrong and people may have a metabolic disorder. Certainly not everything works perfectly, but usually it works pretty well in people or we wouldn't be here. So gene expression, that's the major thing here I wanted to go through. You have questions for me on this? Let's go back through a little of the cancer stuff. Now, I had a question about whether the hallmarks of cancer would be on there. Well, in the first place, there's a difference between cancer and not cancer. Really lovely picture, huh? The hallmarks, I was going to skip these. I'm not going to test you on, say, particular hallmarks of cancer. You should know these aspects that, for instance, cancers involve genetic mutations leading to high rates of cell division. I think that should be fairly obvious. And they're able to then divide no matter whether the cell is being told from outside that whether it's appropriate to divide or not. They're able to avoid programmed cell death. They're able to keep dividing through, in some cases, an infinite number of cycles. Instead of having the telomeres erode off the chromosomes and eventually causing them to stop dividing, they can keep on dividing. And they're able to call for growth of new blood vessels growing into a tumor to let it get bigger. And they metastasize. Definitely, everyone should know what that means. So, just as general concepts, I think you should know these phrases and what they mean, and I'm not going to test you on details of all of that. What I do want to show you is something that does go on here with cancers, and it involves, in some cases, a failure of signal transduction. Now, cells are usually, if they're going to divide, they're typically being told to divide by their neighbors or by hormones circulating in the bloodstream. So imagine you've got a cell that's sending a chemical signal. You have this signaling molecule, possibly a hormone circulating in the blood. It arrives at a receptor. There's a series of signal transduction steps that go on as the signal is heading down into the nucleus and various genes are being activated. So we might have a signaling molecule that is saying, okay, cell, you need to divide. There's a lot of steps involved in cell division. So as the signaling molecule arrives, you end up getting many different signals being passed down through the cell. It's not just a chain like this, but maybe this receptor can set off 10 other proteins to activate different genes that are all going to be promoting cell division. So you have this whole series of steps propagating into the nucleus, activating various genes, switching those on, leading to production of proteins that could be involved in cell division, and many different proteins coming from many different genes, because cell division is a big deal. That takes a lot of proteins to happen. So normally a signal is going to arrive from the outside and set off a series of steps moving into the nucleus, leading to a response. Sometimes the signaling goes wrong, and that's part of what's implicated with cancer. we have various genes, some of them known as proto-oncogenes and tumor suppressor genes. These are normal genes that we're supposed to have, and these are coding for various parts of the signal transduction system. Now, you're supposed to have these. They will then be regulating the pathways that are leading to expression of genes relating to cell division, mitosis, growth of new cells. And if these pathways get corrupted, then you could potentially have a cancer going on. So the proto-oncogenes, these are ones that act to speed up cell division. They're basically the gas pedal on the cell cycle. The tumor suppressor genes are the brake pedal. They're slowing it down. So you can imagine that cells are getting some stimulus to speed up cell division, but there's also inhibition holding them back. Normally, they're going to be divided at the correct rate. If there's more stimulation, then okay, there may be more activity by the proto-oncogenes, maybe less activity by the tumor suppressor genes. In other cases, less cell division, that balance shifts. But if these genes get damaged, then you may get too much stimulation. So you can have damage to these genes. These are the stimulant genes. Those can get damaged in a way that leads to over-stimulus of cell division. If these get damaged, they're the inhibitory genes. There may not be enough holding back cell division. And what happens with cancer cells is that you'll have mutations happening to several genes that are of these types. And collectively, that leads to the activity that we call cancer. So with the proto-oncogene, imagine we've got a gene there that is producing some sort of protein that stimulates cell division. The proto-oncogene is the normal version, but there's different ways that it can get mutated into an oncogene. An oncogene is a pro-cancer gene. It's a mutated version of a normal gene. So you want to have proto-oncogenes. You don't want them to get mutated into oncogenes. Those are the cancerous, harmful versions. And there's different ways that can happen. You could have a mutation within the proto-oncogene that transforms it into the oncogene, and that creates some sort of hyperactive growth stimulant, some sort of part of the signal transduction pathway that is too active, that's basically causing too much stimulation. Alternatively, the proto-oncogene can get duplicated. During cell division, sometimes genes get copied. You can have a mutation where maybe we produce several copies of that gene, and instead of just producing a small amount of a growth factor, we make a lot of it, and now we get too much stimulus of growth. Alternatively, the gene can get switched to a new place, and we had the case with that Philadelphia syndrome, where there is a translocation of genetic material between different chromosomes. That's a well-known cancer where this exact thing happens. You have a gene related to mitosis that is making a protein that would stimulate mitosis. That gene is moved to a different location and it's switched on too often. The gene is normal, but it's sitting next to the wrong control sequence, and so you make too much of the normal growth factor. So you might get either a mutated growth factor or else too much of the normal one. Either way, we're getting about the same effect, too much stimulation of growth. So imagine you have this. We have a signal coming in from outside the cell, binds the receptor. That signal should propagate through a series of signal transduction proteins moving into the nucleus, leading to production at a normal rate of a growth factor. Well, you have here something that is the RAS protein. This one is implicated a lot in cancer. That is produced from the RAS gene. And what can happen is that the RAS gene can get mutated, producing a mutant RAS protein. And this is a hyperactive relay protein that is basically switched on all the time. The RAS protein should only be sending this signal down if it's received a signal from outside the cell. But now we get a mutated version of the RAS protein that is basically switched on all the time, and it's constantly sending a stimulus into the nucleus saying it's time to divide, even if the cell has not received any such signal. So the cell has now decided on its own to divide, no matter what the circumstances are. So that's a mutation involving the RAS protein, and it can be deadly. One of the ones that commonly shows up in cancer. So that mutant gene has been transformed from a proto-oncogene into the harmful oncogene now. There's also the tumor suppressor genes. These are ones that slow down the cell cycle. They're operating with those checkpoints. Remember when we looked at the cell cycle, there's several checkpoints. You've got one going from G1 to S. That is where the cell is being told to start duplicating the DNA. Then there's one at the end of interphase, and there's one in the middle of the mitosis cycle. So you have several points where the cell is being told to stop and make sure conditions are right before going on. For instance, when you're at the end of interphase, about to enter mitosis, the cell has to check and make sure it copied all the chromosomes correctly. If it didn't, it may have to go back and recheck its work, go and fix some things, or possibly commit suicide. You have programmed cell suicide, and a cell that has made very bad copies, corrupted copies of chromosomes, may be told you have to die, and it will do it. But that's under the influence of the tumor suppressor gene, like p53. This one is implicated a lot in smoking-related cancers. They've gone and checked sputum from lungs of smokers who don't have lung cancer yet. But checking sputum, you can get basically DNA from cells in the lungs, and frequently those sputum samples show corrupted versions of the p53. So the toxins that are in the smoke are damaging the p53 genes, and some of those lung cells are then going to be prone to develop cancers because they're no longer doing correct checking of the integrity of the cell at these different checkpoints. So under the influence of a normal p53 gene, you're going to slow down cell division, make a cell really take its time and be sure it did a good job before it divides. But if the p53 gene is mutated and doesn't work anymore, then you're making some corrupted, useless protein, and now the cells are free to divide, even if they duplicated bad copies of the chromosomes. At least one person on one of the posters I know had a chart showing chromosomes that were massively incorrect, where maybe instead of having a normal diploid set, in some cases there were 4 copies of one of the chromosomes. It was just an absolute mess. And that's happening basically because of the failure of genes like p53. Normally a cell like that would have been just, auto-suicided, would have committed suicide by apoptosis. But once we've gotten into the cancer situation, that's no longer happening. So we get an interplay of these kinds of mutations, speeding up cell division and inhibiting any sort of breaking process on it. And usually it takes several such mutations, all occurring in the same cell, to lead us to a cancer. So we can have something like maybe a healthy colon, healthy at one point. But let's say now we have one of these mutations happens in a cell. A single cell has had an oncogene develop by some mutation to the normal proto-oncogene. We end up then having this accelerated cell division. We produce a mass of cells that all share that mutation. And years may go by in the case of colon cancer, but you've now got a mass of a large number of cells that any one of which might develop a second mutation. So now within this initial mass of cells that all have the one mutation, a second mutation happens in one of those cells. And now we get much more growth occurring, and we get something that is macroscopic, a polyp that might be, say, that big, with millions of cells, and they all have both mutations. Within that polyp, if you wait long enough, a third mutation may happen in one of those cells, and now it's exploding into a cancer. So we may go from a few hundred cells to a few million there to billions. And it's described sometimes as tumor evolution. Essentially, there is a survival of the fittest process going on, where a cell that is able to more and more escape the controls that hold down cell division is able to produce more offspring. You think about it in nature with evolution, part of the way that a species wins is by, or an individual wins, is by making more offspring. Essentially, that's what the cancer cells are doing, up until the point that they kill the whole host and everyone dies. But up until that point, it's great for them. So, what'll happen is you'll go from this, let's say that's the polyp stage, precancer, and we can get this taken care of. We can fix that without too much harm while it's still at this stage. This has not exploded and destroyed the person's life yet, but there it has. That's, this is going to be probably your stage 4 colon cancer. It's definitely spreading and escaping from the area. Here, it's not a cancer at all, not yet. And we could remove that and take care of the problem. At this point, things are much worse. This person's life will never be the same again. So they'd have to do massive surgery. The person might be wearing a colonoscopy, a colostomy bag for the rest of his life and so on. And who knows how long that life would end up being. Here they can take it out and you're fixed. So it's definitely a tragic thing. This cancer, this particular one, is slow to develop, and we can deal with it, but it's hidden and people are embarrassed about it, and so often it goes overlooked. If this was on the outside on your skin, you know, everyone would get it taken care of and it would be okay. So, has anyone here had a colonoscopy done? Yes, you're all too young. Well, you'll probably have to have it done by the time you're 45 or so. The rates of colon cancer in younger people are rising. And they used to say 50 for getting your first one done, but recommendations are going down now because they're getting cases where maybe a 27-year-old will have the symptoms of colon cancer and be seeing her doctor. The doctor will overlook it or just think, oh, it's something else. Even if he saw a 60-year-old with the symptoms, he would automatically think colon cancer, but he just doesn't think of it with a young person. So sometimes things go overlooked for too long. So you'll probably all have to get one done when you're, 38 or so, something like that. Okay, probably not as soon as that. But the colonoscopy, I did have one done when I turned 51. I'm due for another one about any time now. I didn't really want to get it done. I had the general male attitude that, oh, I'm indestructible, nothing will happen to me, that kind of thing. And I turned 50 and my wife started nagging me about getting one done. Said, okay, you're old enough, you gotta get it checked out. And, I didn't want to. And so I was really whiny about it. Saying, I don't want to, like that. But she pressed me on it, and I'm glad she did. So I finally scheduled the whole thing, and it's kind of complicated. You need to have someone arrange to drive you home from the clinic because you're kind of sedated and not fully with it when you come out of the procedure. So it's not just a matter of going in the doctor's office and leaving. But you have to get yourself cleaned out before you go in. They want to pick up these little things like polyps and not get distracted by food particles. And so you go on a special diet where you're not eating anything that's granular, that would have like seeds in it, something that might get hung up here in one of these folds and trick the technician, because he's going through you with this scope and he wants to make sure that anything he sees is going to be like that. So you go on this rather bland, boring diet for a couple of days, and then the night before the procedure, you go to the pharmacist and pick up this gallon jug that has a powder in the bottom, and it's a laxative. And they asked me, what flavor do you want? They had a couple of choices. I think I picked the lemonade Crystal Light version. And you put in a gallon of water and you start drinking this stuff, and very quickly you end up parked on the toilet and you're just going and going and going. You drink this stuff and 5 minutes later you're back on the toilet again. And I probably had to use it about 20 times. Until it's getting about 10 o'clock at night, and I could tell I was totally cleaned out. There was still about this much of the liquid, and I just threw it aside, just thought, oh, the hell with this. That has to be enough. And she took me into the clinic the next morning, and they give you a sedative to get you sort of knocked out. And they take a tube that is flexible, and it's about this long, and they bring it in, they bring it up through the sigmoid colon, up the descending colon, across the transverse, and down the ascending colon. And so it takes about that much of this piping that's very flexible to snake all the way around through the large intestine. And it has a camera. So some technician, that's his job every day, is to look at this kind of thing. Lovely. And it has the camera, and it also has a tool where they can remove stuff like this, so they can bring specimens out without having to do a big, major surgery. It's this painless removal of little polyps, and they bring them out for study. All of that's going on, and you're sedated, and that's kind of nice, actually. I really didn't want to be aware of all this at the time. And I'm waking up, and they say, hey, we found a polyp. Do you want to see it? Or do you want it? And it was only about 1 millimeter or so in size. This one is 17 millimeters, so mine was more like that. Tiny, tiny polyp. They asked if I wanted it, and I went, no, you keep it. But I wish I had kept it, because I would pass it around in here right now. You'd all get to look. I just wasn't thinking clearly enough at the time to get my sample. Oh well. So they found a little polyp and took it out. And that's the kind of thing they can go and intercept as long as they do it. And if they don't check it, it turns into this. So pretty hideous. So just a brief summary of cancers in the United States. Well, lung cancer is the most common, but prostate and breast are roughly as common too. Of these three, which one would probably be the biggest killer? Because this is looking at number of new cases. It's not saying how many people died. Which of those top three would probably be the biggest killer? Yeah, lung. Most people die with lung cancer, although I have an aunt who has survived a long time with it, but most people don't. Prostate, most people live. Breast cancer, most people live. So even though we're getting about the same number of cases, lung is certainly the worst one overall. Then notice we've got colorectal cancer and a lot of other types through here. What appears to be the top risk factor? Tobacco smoke, and that makes sense, of course, with lung, but bladder cancer, kidney, it's not where you're putting in cigarettes. So why would kidney cancer be influenced by tobacco smoke? Yeah.
Your kidney and the process
waste.
Smoke.
Yeah, you're getting all of this poison from the smoke in your lungs. It's getting into your bloodstream, and what are the kidneys doing? They are filtering the blood. and they're exposed to it heavily. And then it passes down to the bladder, and it's the same deal. Like, you've got basically urine contaminated with poison sitting for long periods in the bladder and triggering then bladder cancer. Other parts too, like the pancreas, maybe not so obvious why that would be the case, but tobacco smoke in general is poisonous, and it's causing harmful effects in many systems. Yes?
So where it says like breast cancer, estrogen,
Yeah.
there's estrogen.
So it's a
does it mean,
hormone, it's a hormone.
Well, yeah, but does it mean like an excess amount of estrogen,
I
or just like?
think it's related, and I would have to look more at that. Some of these are obviously tied to gender.
Yeah.
Hardly any males get breast cancer. I'm sure no women get prostate cancer. Some other things are related to, for instance, ethnic background. In other words, things we have no control over. But I think it's probably most productive to look at things that we do have control over. Melanoma, obviously, is related to UV exposure, and there are things you can do about that. There are other types of cancers, and some of them related to viruses. That is one thing. Cervical cancer, also oral cancer. You have some cancers related to the virus, HPV, and we have a vaccine against that. So basically, any part of the body that gets involved in sex is subject to HPV infection, and it's known for a long time to cause cervical cancer, also cancer of the penis, and anal cancer, and mouth cancer. Again, these all get involved in sex. And so HPV vaccine is a smart thing to do to protect yourself or protect your kids from it. But no, more than anything, it's tobacco. So if you're looking for a single thing to improve your health, throw tobacco away or never pick it up. So this is, I think, a worthwhile thing to do, just to summarize what you guys were already seeing with the cancer topics. And a lot of those posters look nice, by the way. I should have those ready for you to pick up on Monday. And I was asked about the last quiz that you took a few days